Hello grade 10 students. In this video we're going to talk about our new lesson, Transgenesis, and also we're going to talk about transcription and translation and what's the difference between both of them. First of all, let's start with transgenesis. What is transgenesis? Transgenesis, it's a process by which a gene is transferred from one species to another species, and this gene will be expressed in the other species. Well, the meaning of transgenic. Transgenic is the organism that carries and expresses a gene from different species or from another species. So as I said, transgenesis is the process where a gene is transferred from one species to another, and this gene will be expressed in the later one, in the other species. Transgenic, it's an organism that carries a gene from another species. Let's move to this experiment. We have here removal of the eggs from a mouse, injection of the human insulin gene. We inject human insulin gene into the mouth in the form of DNA inside the male pronucleus, and then it's injected into the female pronucleus. A number of altered embryos were transferred into the uterus of a foster mother, as shown here. Then we have the birth of 3 mice. 3 mice, 2 of them, they showed negative result. There is no insertion of the DNA fragment, whereas 1 of the 3 mice shows that there is a positive result. We call it a transgenic mouse. So after the injection of the male pronucleus of the mouse that is already injected by human insulin gene, so here we inject a gene from another species, from a human to mice, and this is injected by a human insulin gene in the form of DNA into a female pronucleus, and we transfer them into the foster mother. The transfer of the altered embryos, they are transferred into the uterus of the foster mother. The result was we have 2 mice with negative result. There is no insertion of DNA fragment, whereas 1 of these mice showed a positive result, meaning that it's called a transgenic mouse. It showed a positive result. So this indicates that if you want to interpret, this is analysis. If you want to give indication, this indicates that the human insulin gene that can code for the human insulin hormone inside the positive mouse, the transgenic mouse. So describe, eggs are removed from a mouse, then a human insulin gene in the form of DNA is injected into the male mouse pronucleus, which is injected into the female pronucleus. Then number of altered embryos, they are transferred into the uterus of the foster mother after the birth of 3 mice, and searching for the human insulin gene in their blood. 2 of these mice showed negative result. There is no insertion of DNA fragment, whereas 1 of these mice showed a positive result, and we have the insertion of DNA fragment, and this is qualified as transgenic mice. Okay, let's move to interpret. Interpret including analyze plus indication. So after injection of the male pronucleus of the mouse previously injected with a human insulin gene in the form of DNA into a female mouse pronucleus, and transferring the resulting altered embryos into the foster mother, 2 of the 3 showed negative result and 1 showed a positive result, which is qualified as a transgenic mouse. So what does this indicate? This indicates that the human insulin gene in the form of DNA in mice can code for the production of the human insulin hormone in transgenic mice. So what can we conclude here? can conclude that a gene made up of DNA can code for production of specific proteins. Let's move to chromosomes. A slide about chromosomes. A chromosome at metaphase is made up of 2 chromatids. That's why it's seen well at metaphase. Then the subset that make up the chromosome is called the chromatin. Chromatin is divided into DNA composed of the nucleotides A, T, C, G. So DNA is composed of different nucleotides A, T, C, and G. These A, T, C, and G, they are called the nucleotide. Every 3 nucleotide, they are called codon. For example, if I have A, T, C, for example, then I have C, G, C. So ATC is called a codon. Codon, it has another name, it can code triplet. So 3 nucleotides together, they are called codon. And later on, they will code for an amino acid. And we will discover this in the coming slides. So you have to know that we have 4 nucleotides, A, T, C, G. You have to know that every 3 nucleotides together, they form what's known by a codon or a triplet. CGC also is called a codon or triplet. You have to know that A and T, they are complementary. And C and G, they are complementary. Chromatins divided into proteins, they are composed of histones and non-histones. DNA, by the process of transcription, they can code for mRNA, messenger RNA. And mRNA, by the process of translation, they can code for polypeptide. So you have to know that we have 2 processes here. We have transcription and we have translation. Transcription is the process where we have production of mRNA from DNA. Sure, we have some enzymes, they should interfere. We have an enzyme called helicase and another enzyme is called RNA polymerase. Whereas translation is the process where we have production of polypeptides by the process of translation starting from mRNA. So let's start with transcription. What is transcription? As I said, transcription is the process by which the mRNA is produced based on the given sequence of DNA. It occurs inside the nucleus. What is the fate of produced mRNA? The mRNA undergoes mutation, some changes, and it's transported from the nucleus to the cytoplasm. So DNA produces mRNA by the process of transcription by the help of helicase and RNA polymerase. So what do? Helicase, as you know that DNA is double stranded, so helicase unwinds the DNA of the gene coding for the protein. RNA polymerase, it has another function. It synthesizes the complementary strand for the transcribed strand of the DNA. So as you know, you have 2 strands. We're going to talk about them also. Consider the sequence of the DNA molecule shown. This is the sequence of the DNA molecule. So you have to be careful which sequence I will give it to you. First, we have this sequence GCA, TGC, ATC, TGC, CAT, and TG. First of all, you have to know that every 3, I said they are called codon or triplet, every 3 nucleotide. This is the non-template. Non-template, we can call it the non-transcribed. And it's called the coding. Okay? They said part 1, write down the sequence of the mRNA molecule shown. We have to write down the sequence of the mRNA. Before we write the sequence of the mRNA, we have to be careful to symbols. First of all, they gave us the non-template. Non-template is called non-transcribed and it's called the coding. So non-template, non-transcribed, and coding. First, let's write down the complementary sequence for this strand. The complementary, it's the opposite. It's called the non-coding. It's called the transcribed, and it's called the template. So let's write down the complementary. As I said, A and T, they are complementary, and C and G, they are complementary. So G should face the complementary. The complementary for G is C. The complementary for C is G. The complementary for A is T. So you have to write them under each other so you don't have mistakes while writing them down, okay? T, the complementary is A. G, the complementary is C. C, the complementary is G. Then A, the complementary is T. T, the complementary is A. G, C is the complementary, it's G. And then we continue. T, the complementary, it's A. G, it's C. G, C, it's G. C, the complementary, it's G. A, it's T. T, it's A. And then we have T, the complementary, it's A. And G, the complementary, it's C. Some of you are going to ask me, this is only two. This has a complement, sure. So it's, for example, we get only the portion of the sequence, okay? So this is the complementary strand. This is the complementary strand. How can we determine the sequence of the mRNA molecule? In order to determine the sequence of the mRNA molecule, you have to use one of these two ways. Either you can use the coding, which is the non-transcribed and the non-template, or you use the non-coding, the transcribed, the template. You choose which one you want to use. But you have to be careful to a specific note. In the mRNA, T is going to be replaced by another nucleotide which is called U. So T is replaced by U. You can use either the non-template or the template. Let's use the non-template, the one in blue. Let's use this sequence. If I want to use the non-template, the sequence, it's the same. The mRNA sequence is identical. mRNA sequence is identical to the non-template, but T should be replaced by U. Like the case, GCA, there is no T, so we put it GCA. TGC, we have T, so T should be replaced by U. UGC. ATC is going to be AUC. TGC is going to be UGC. CAT, CAU, and UG. This is the sequence of the mRNA. Here we use which one? The non-template. So the non-template sequence is identical to the mRNA sequence, but the T should be replaced by U. But if I use the non-coding, the transcribed, the template one, this one, it should be complementary. But the T, it should be replaced by U. For example, C, the complementary is G. G is the complementary, it's C. T, the complementary, it's A. A, the complementary is T. But in the mRNA, there is no T. The T should be replaced by U. So we have to put U. U. C, the complementary is G and C. T, the complementary, it's A. A, the complementary, it's T. But there is no T. You have to be replaced by U. C. A, the complementary, it's T. But there is no T. It should be U. C, it's a G. G, it's C. G, it's C. T, it's A. And A is going to be T. No T. It's U. A is going to be T, but there is no T. U and G. Here, if you notice, we have the same sequences. Either we started with the non-template, which is the non-transcribed, the coding, or we started with the transcribed, template, the non-coding. Okay? How you may relate mRNA to the template, to the non-template. As you see here, the non-template is identical to the mRNA, but the T is replaced by U, while the template it's complementary, and the T is replaced by U, as you see here. So this is the sequence, and this is the notes. The mRNA, you have to refer to this note. The mRNA is complementary to the template, and the mRNA strand is identical to the non-template, and the T is replaced by U. Here, the notes about the functions of both helicase and RNA polymerase. Helicase unwinds the DNA of the gene coding for the given protein. RNA polymerase synthesizes the complementary strand for the transcribed strand. So now we move for translation, which is the second step of the protein synthesis. First, we started with the transcription. It's the movement of the DNA level to the mRNA level by the help of the enzymes helicase and RNA polymerase. Translation is the process by which the RNA in the cytoplasm will code for the production of proteins. What will happen for these proteins? What is the fate of these proteins? Some of them they remain in the cell, and others they are exported out of the cell. Given the following coding DNA strand, they said it's the coding. So it's the template, the non-transcribed, the non-template, it's the coding. Give the sequence of the transcribed, non-transcribed, non-template coding. So the transcribed is going to be the complementary one. So the complementary AAA, the complementary is going to be TTT, CCC, GGG, GGG, CCC. CAA is going to be GTT. TCG, T complementary, it's A, C, it's G, G, it's C. So this is called the transcribed. Specify the sequence of the mRNA. The sequence of the mRNA, we have the coding strand. So the coding at the same sequence, but the T is replaced by U. So AAA the same, CCC the same, GGG the same, CAA the same, TCG, T is replaced by U. So it's going to be UCG. Okay? And the part 3, specify the sequence of the amino acids found on page 68. So here, what I have said before, the sequence of the mRNA. How can we know that? First, AAA, CCC, GGG, CAA, UCG. Let's start first with AAA. This is the genetic code table. AAA. The first letter is A. The second letter is A. The third letter is going to be what? So AA. So let's see here. AAA. It's LYS. This is the amino acid. Second one, it's CCC. So the first letter is C. So the second letter is C, and the third letter is C. So CCC, protein. Let's move now to the third one, GGG. The first letter is G, second letter is G, and the third letter is G. So here it is GGG, GLY. Okay, and let's continue. So we have, other than these, GGG, and we have CAA, CAA. The first letter is C, second letter is A, GLN. This is GCAA. And we have the last one, UCG. U, first letter is U, second letter it's C, this is the second letter, and the third one is GSER. So this is the sequence of the amino acids by the help of the genetic code table. Sure, you can't study this, and you don't have to study unless you have to know that we have 3 stop codons. These 3, you have to know them. You have to know that UAA is a stop codon. Whenever we reach this will cause a mutation. We'll discuss this next year, inshallah. So UAA, it's called a stop codon. UAG is called a stop codon, and UGA is called a stop codon. So these 3 codons, UAA, UAG, and UGA, you have to study them. Okay? UAA, UAG, UGA. It's easy. You have to study only these 3. you have to know that they are called stop codon. So these are the notes required for the sequence